So, good afternoon, everyone. Very happy to see as many people online. Welcome to this webinar of the German Society of Gene Therapy. My name is Toni Katzomen. I will be the host of this webinar today in my function as past president of the Society. And it's a great pleasure to also welcome our speaker today, Julia Peterson. And just give me a minute to introduce her to you. Julia was born in the United States. She did her undergraduate degree in Michigan before she came over to Europe. And then she moved to Utrecht University, where she got her master's degree in pharmaceutical sciences, and then decided to come to Germany. To the DKFZ, the German Cancer Research Center, where she joined Richard Hartbottle's lab, starting to work on iPSCs, and this is also what she's going to talk about today. And maybe we start sharing the slides.
Yeah, sure.
Then I can give you some housekeeping remarks. Exactly, I think we would like to make this as interactive as possible. So you can send us your questions on the chat. You can see the instructions here. So just whenever you have a question, whenever anything pops up in your mind, just write it down in the chat. And then you just have to be aware that these live messages are not private, so everyone can see them. And I will then forward the questions at the end of Julia's talk to our speaker, where she then can answer them. I think this is more or less everything I wanted to say. And I'm very happy to hand over to Julia, graduate student, as I said, at the DKFZ, who will give this nice presentation today.
Okay, so hi, everyone. As Tony said, I'm Julia. Thank you so much for attending today's webinar. Today, I'm really happy to share with you some data we've collected towards the development of an off-the-shelf T-cell immunotherapy. And for that, this presentation is primarily focused on the first aspect of that, which is the HSPC differentiation. So in order to produce the cell therapy, we're utilizing urinary-derived iPSCs, as well as a special genetic modification tool. So there are various genetic modification tools one can use to genetically modify a cell. There are viral vectors, there's transposons, and there's CRISPR-Cas. Our lab is specifically focused on using SMR DNA vectors. Now, what are those? And how does it compare to, let's say, a classic semi-randomly integrating viral vector? So with a randomly integrating viral vector, what happens is that the provirus is introduced into the cell, which then semi-randomly integrates into the genome. And because of that, we're having long-term persistent expression. With the system we're using, SMR DNA vectors, what we have is a circular piece of DNA, so plasmid, and we introduce that into the cell, commonly by using electroporation. And what happens is that, unlike a lentiviral vector, it persists episomally. So it's not integrating into the genome, but like a lentiviral vector, we have long-term transgene expression persisting. So this is a bit rare. It's a bit uncommon because most people would correctly assume that when you're using an episomal system, episomally maintained system, you eventually have transgene expression lost. So this isn't happening with a particular subset of the cells we modify using SMR DNA vectors, because we're using a special sequence of DNA called scaffold matrix associated region. So that's what SMR stands for. So these sequences, they're naturally found in the genome of, oh, I just realized no one probably can't see my whole slide. See if I can, 1 second. Oh, okay, well, Okay, well, you can see the majority of my slide there. I don't know how to fix that. Okay, so SMAR sequences, they're found in the genome of various species. So here, particularly, I'm focusing on the human genome. And what is found is that in areas of the genome where there's a high prevalence of SMAR sequences, there's an upregulation of transcription. And also it's found that these sequences, they're used to anchor the genomic DNA to the nuclear matrix. And this is important, and it's been shown that this anchoring allows the organization of the chromatin into a 3D structure. And by doing that, it's regulating transcription and also the replication of the genome. So when we're using plasmids, which contain SMAR sequences within them, we also find that they associate closely to these attachment sites where the chromatin is attaching to the nuclear matrix. And additionally, we find that subunits of the origin of replication complex are associated with that SMAR sequence within the plasmid. So there's literature that's showing that. And SMAR sequences, they have been found in mammalian origins of replication. So this naturally would, in a way, make sense. So seeing that, these associations with the subunit, it kind of supports the hypothesis of why the vectors are being maintained by kind of hijacking this origin of replication complex and replicating along with the genomic DNA as the cell divides. So how do we know and prove that the vectors are persisting episomally? So there are various tests one can do for that. So here particularly, I'm showing two different assays. One is rolling circle assay, and the other is southern blot. So I'll primarily focus and describe the rolling circle amplification. So M here is our ladder, and then we have our controls on the left. So this is our plasmid DNA that had rolling circle amplification performed on that. So it's just essentially a PCR that should be only amplifying circular DNA. So we have that. After the amplification, we have to cleave it. And we can see after we run it on a gel, we're having one band present. So this is our control to say, this is where we would expect to see this plasmid when we perform this assay. Now over here on the right, we have the genomic DNA of cells that were modified using our vector system. So WT wild type, that's non-modified cells. And here we see the presence of no bands predominantly. And we have our down arrow here, which matches with our down arrow control here. And also same with the up arrow. And within these, the genomic DNA of these cells, we can see a band present. So it's indicating we do have circular DNA present still after establishment using the system. Now with the southern blot, it's a different technique, but how you analyze your control to your modified cells, it's essentially the same, looking at the bands. So our lab has made various improvements to these SMAR DNA vectors. So we've done this by making alterations to the promoter, so switching CMV to COG. We've also removed an enhancer, UCO, and replaced it eventually with element 40. And we've made changes to the production backbone. So rather than using a classic kanamycin backbone, we're using an RNA out backbone, specifically from Nature Technologies. And here we can see by making these changes, it's led to improvements in the efficiency of the establishment. So we can see here by looking at these graphs over 90 days, we can see with our older versions of the SMAR, we have a lower establishment efficiency, creating lines that are expressing our transgene of interest. So in this case, we were using a reporter gene, GFP, and we see very low presence of this expression, but with our newer versions, we have a high expression and presence of that. And we can see this also here represented in a line graph. These are our newer versions. And these was also associated with a more intense expression of our transgene, which we can see here with MFI. So we've also made changes to the SMAR sequence. So we've taken SMARs that were derived from a different area of the genome. We've also made changes to it based on the size, and we've implemented specific features to it as well. And we found that these additional changes have also improved the establishment efficiency of the vector system. We also found that these modifications, some of them at least, have led to fewer differentially expressed genes, so indicating less likely to affect the functionality of the cell type we're using it to modify. So PSMAR here, this is using a classic kanamycin backbone, and we're comparing that with NMAR, which was just using that minimalized backbone RNA out from Nature Technology. And we found this RNA out backbone has led to that fewer differentially expressed genes, and it wasn't suggested to be associated with the intensity of the transgene's expression, which we show here over in A, because we didn't find a significant difference between these two groups. And actually the mean expression intensity for our NMAR line was actually higher than PSMAR. So yeah. Though when we looked at the overall messenger RNA profile of the cells, we saw a closer relationship with our SMAR DNA vector modified lines and furthest from non-modified. But when we compared that with cells that were modified using a lentiviral vector, we actually had a closer relationship with the NMAR in the mock and the furthest from the lentiviral vector. So we've used these SMAR DNA vectors for various applications. One of them is for T-cell immunotherapies. So here we're doing a proof of concept showing that T-cells that are modified to express CD19 using our SMAR vectors, they were able to treat mice that had tumors implanted inside them. And it was the treatment regimen, how effective the treatment was very comparable to using T cells that were modified to express CD19 using a lentiviral vector. And we can nicely see this as well depicted in this line graph here, where we have a significant reduction in tumor size compared to non-treated. And it's very comparable, the treatment, if you're using SMAR or lentiviral vector to modify the T cells. So we've also used the system to genetically modify human iPS cells. So here we see the phenotyping of these modified cells, and we showed that they could produce alkaline phosphatase and express the pluripotency markers. And it was comparable to non-modified lines. The only difference really was in the expression of our reporter gene. We also showed that these modified iPSCs were functioning like an iPSC still because they successfully underwent trilineage differentiation. And then we've looked more closely at how the cells were able to maintain transgene expression, because this is really important, specifically for the project I'm going to be sharing with you guys today, because we want to see how transgene expression is affected after going through this differentiation process. And we showed that when we're using SMAR DNA vectors, when we have non-modified, which is represented in black, and we compare that to our differentiated cells into the 3 different germ lines, we can see that all of them on average were maintaining, so 90% of the cells were maintaining transgene expression, which is a really nice indication for us. And additionally, we looked at how intense transgene expression was occurring after differentiation. And we see on average, the mean averages are being reduced with that. So it's something really important to keep in mind that for our system, in order to produce these iPSC-derived cells, we probably really want to start off with something that's really intensely expressing our transgene. So there are no, I would like to point out as well the statistical analysis with this. So even though the mean average, the mean intensity was reduced, we didn't see a significant difference compared to undifferentiated with our newer versions, while we did see that with our later versions, also for the percentage of cells maintaining transgene expression. So this is now where my project kind of comes in. So it was, can we kind of combine these two aspects, T-cell immunotherapy and iPSC cell therapies? But why? The question is why? What is the benefit of using an iPSC-derived T-cell therapy over a classic cell therapy? Why would we want to mix it up? So one of the biggest benefits, in my opinion, is that it has the potential to reduce the cost of current cell therapies. So right now, due to one of the major reasons why they're so expensive is due to this high personalization. So if we can kind of reduce the personalization a bit, this could potentially help reduce some of the costs associated with it. So when using an iPSC-derived cell therapy, we can produce the cell therapy in bulk, and then we can then aliquot these cells, and then it's essentially therapy that can be used to treat multiple patients. So instead of just one, it can treat multiple. And there are different ways you can propose which donors you need to use, because obviously you do need to still have some personalization to a degree, because we want patients to be matched with an HLA match and treated with the correct CAR. So I would propose starting using donors that are HLA homologous. So then it's been shown in literature that that can treat a larger population. So yeah, so one of the biggest benefits, in short, is that you can produce a bulk amount of cells that can be used to treat multiple patients. And then additionally, it can be an option to treat patients more quickly. So instead of having to wait 2 to 8 weeks to have their cells processed, it provides the opportunity for them to be treated more quickly because the cell therapy is already prepared. Additionally, it can be derived from healthy donors. So one issue with cell therapies, and this is really common for pediatric patients, is that doctors are not able to collect enough cells to perform the cell therapy. But if we're producing it from a donor where we can essentially expand indefinitely, there's a limit, but yeah, indefinitely, we can have enough cells to treat these patients. And additionally, patients we're taking T cells from, a lot of the times, for immunotherapy specifically, they're sick. So the functionality of their T cells might also not be as strong. So if we're starting off from a healthy donor, it may have actually a stronger effect for the treatment. And then lastly, something more for a regulatory aspect is that we can have a standardized therapy that could follow more easily a centralized model, which has been kind of debate for cell therapies, and because it's a little bit difficult because how personalized they are, to take the cells from a patient and send it off to a facility and then send it back. This is the kind of the concept of the centralized model where everything's produced at one location. And they've been proposing to do decentralized models where everything's produced at the location where the cells are collected. But there's some questions about how can we keep it standardized, the therapy. So in this case, because everything could easily be produced at one location, I say that it's a standardized therapy that could more easily follow this method. Okay, so how can one create an iPSC-derived T-cell therapy? So for this proof of concept First, we want to show that we can produce iPS cells. And we were producing them using, so we were taking cells from the urine. And we felt that this was a nice way of doing it because we could potentially have more donor participation because we're using then a non-invasive method to collect the cells. So this was all set up by a previous PhD in our lab. Her name is Manuela Urban, and she showed that you could collect the urine from a patient, take the cells from that, plate them in the proper medium conditions, and then grow out and obtain these urinary stem cells. You can then introduce reprogramming vectors into them, and after they've been in culture for an appropriate amount of time with the appropriate conditions, you end up with these urinary-derived iPSCs. So Manu has shown that these urinary-derived iPSCs function like an iPSC because they had successfully undergone trilineage differentiation. So here we have IF images of those 3 different germ lines they were differentiated into. And she also showed they're phenotypically like an iPSC because they were able to produce alkaline phosphatase, and they express pluripotency markers Lin28, NANOG-G, Tr160, and OCT4. Okay, so the next step for us is that we were really keen to investigate various features that would give us the strongest chance for the genetic modification for creating these established lines. So for that, we were looking at 3 different promoters, 4 different SMARs, and 2 production backbones. I've already kind of explained a little bit about the benefits between these in the introduction, so I won't go too much into it with that, but with the promoters, here we have transfection efficiency in human iPS cells. So we were looking at COG, short elongation factor 1, and short elongation factor 1 with a chimeric intron. So we had the highest transfection efficiency when using COG and the lowest with our short elongation factor 1 promoters. When we looked at the associated viabilities with that, we had one of the lowest with COG and one of the highest with the short elongation factor 1, which in a way is a little bit expected because the viability and transfection efficiency, a lot of the times they're a little bit negatively correlated. However, with the short elongation factor 1, with the chimeric intron, we had both one of the lowest transfection efficiencies as well as one of the lowest viabilities. And we also were not able to successfully establish a line using that promoter, while we were able to do that with the other 2, and we had the highest establishment efficiency using the COG promoter. To then get an indication of the immunogenicity different vector features are causing, we used human fibroblasts as a model. We introduced the vectors into them, and then we assessed the amount of INF beta that was being secreted from the cells. And again, we also found that the addition of a chimeric intron to short elongation factor 1 led to an increase in the mean concentration of INF beta. And we also saw that when we were using a completely different promoter, when we added a chimeric intron to that, we also had an increase in the mean concentration of INF beta. We then looked at long-term retention. So for us, this is really important because we want to have a therapy that can be expanded for a long period of time, treat many patients, and also it needs to go through a really long period of differentiation to obtain our cell type of interest. We really want to make sure that transgene expression is persisting throughout this process. So for that, we were using Jurkat cells. And after the line was established, we kept the cells in culture for 128 days. And we found that after day 64, we had a significant drop in the percentage of cells retaining transgene expression when we were using COG. And we didn't have this any statistical differences when using our short elongation factor 1 promoters. So because of that, and because of the immunogenicity that's associated with that chimeric intron, we decided to continue on with short elongation factor 1. We then compared SMARs. So this is the transfection efficiency also in human iPS cells. And we didn't see really any differences between the 4 different SMARs we were looking and assessing. However, we did see a trend with the associated viabilities in a way, kind of from going from SMAR 1 to 4, we had a trend of increasing in the viability. So the difference between SMAR 1 through 4 is that SMAR 1 and SMAR 2 through 4, they're derived from different genes. Yeah, they're derived from different areas of the genome. And SMAR 2 through 4, it's getting smaller in size. So we were trying to find the most functional aspect of that gene region. And so we're kind of narrowing down the sequence, but we've shown that this smaller sequence, it did still had functionality similar to the larger ones, which is why we're screening it here. Looking at our establishment rates, so the number of cells, the subset of cells that were retaining vectors long-term, we were able to create lines with all the SMARs, but we had the highest efficiencies using SMAR 1 and SMAR 2. Now, looking at immunogenicity, we also saw a similar trend like we did with our viabilities. So we had the mean concentration of INF beta there was a trend of it being reduced going from SMAR 1 to 4. Then looking at long-term retention in Jurketts, we found a significant drop from day 0 to day 128 for SMAR 1 and SMAR 3. We did not see this for SMAR 4. And I would like to point out, though, even though we had a statistical difference in the drop, at 128 days, all of our SMAR sequences were maintaining nearly above 90% of the cells. Nearly 90% of the cells were maintaining transgene expression, which is really cool. But nevertheless, because of this long-term retention we were seeing with SMAR 4 with no statistical difference and the suggested lower immunogenicity, we decided to continue using SMAR 4. Now, really quick with the production backbone, we are comparing a classic ampicillin backbone to the RNA out. So this is, again, transfection efficiency in human iPSCs. We can see when we're using this RNA out backbone, we had an increase in transfection efficiency, an increase in the associated viability, and we also were able to establish lines with it when we weren't able to do that when using the bacterial backbone. So in all, we decided then to continue with a vector containing these features I just mentioned. And we're using this vector now to modify our urinary-derived iPSCs we just developed and differentiate those into T cells. But the first step before we do that is that we need to differentiate into an HSPC. So prior to T cell differentiation, the cells must first go into an HSPC. So for that, first we have to modify our lines. So we were comparing this with a lentiviral vector that had the same expression cassette as our SMAR DNA vector. So this is 3 days after the addition of our DNA liposuction complexes. We had around 80% of the cells expressing D-tomato. And when we compare that when we were using the lentiviral vector method/system, we had around 33% of the cells. So for that, we applied the virus, we removed it 24 hours after, and then 3 days after addition, just like what we did with the SMAR vectors, we assessed the percentage of cells maintaining transgene expression. And we can also see here in B, maybe a little bit more better, that we had a higher intensity of transgene expression occurring with the lines modified with the SMR DNA vector compared to the lentiviral vector. And we could visually see this is very apparent also with our microscope images here when we compared our control to the SMR to the lenti. So also with our microscope images, we get an indication of the viability. So how these systems are affecting cell viability. It's because because this is an adherent line, because the cells are adherent, we're aspiring a lot of the dead cells. So assessing the percentage of alive cells using FACS is not so accurate. So here with these microscope images, we have the cells seated at the same density, and microscope images are taken 3 days after. So we can kind of get an indication of how the viability is being affected by seeing how many colonies we have visibly present. So with the SMR DNA vectors, we had a lot of colonies similar to the control, a little bit less, but similar. And with the lentiviral vectors, we had very minimal colonies. So this was very suggested to really have an effect on the viability. So next, we created established lines. So here's with the SMR DNA vectors. So 11 days post modification, we had a very low percentage of cells, which was expected. And we seeded these cells at a 3% density. So 3% of the cells were positive for the transgene. And 6 days after this, we had a drop then to 2%, so 1/3 loss of the cells. But when we sorted those out, we had essentially transgene expression maintaining. So we did do 2 additional sorts just because I wanted to follow a classic method that we use within our lab to establish SMR DNA vector modified lines. And after that final sort, we then kept the cells in culture for 1 month to really determine how established the line is. And we saw minimal losses. So the majority of the cells were expressing transgene still for 1 month after our 4th sort here, which is really good. It indicates we had an established line. For our lentiviral vector modified iPSCs, I was following a classic method for the lentiviral vector. So for that, we had 1 week post modification, around 1.5% of cells that were still expressing the transgene. And then 1 week after that, we had a drop to around 60%. And we again also, after this final sort, assessed the percentage of cells that were still expressing our reporter gene 1 month after. So because we sorted out the cells on day 14, we would expect to see 100% still maintaining 1 month post if we were not having a loss. But in this case, we actually had a drop down back to 60%. Comparing the mean fluorescence intensity, we also saw a stronger expression of the transgene with the SMR DNA vector modified lines, which isn't so surprising because we saw a similar data when we were performing the transfection efficiency. So to get an indication of how this genetic modification is affecting the cell's functionality, we assessed the phenotype of the cells. And by comparing that with the control, we can kind of get some indication of the functionality, simply because the control has already undergone a trilineage differentiation. So if the phenotype is looking the same, then it gives some indication that we don't have so much alterations happening. Obviously, there's more tests that need to be done to really prove that, but just to get an idea. And visually, we saw no differences in the production of alkaline phosphatase or the expression of Lin28, NANOG-G, TRAL160, or OCT34. The only difference was really in that transgene expression. We then took these lines and we underwent we took them and we differentiated them into HSPCs. And for that, we were using the STEM diff hematopoietic kit from Stem Cell Technologies. So on day 0 here, these are our plated iPS cells. And day 12, this is after the cells have undergone that differentiation. And visually, we can still see that we have transgene expression. When we phenotype these cells, we found that we had between 42 and 50% of the cells for these lines to be double positive for CD43 and CD34. So CD43 gives an indication that our pluripotent cells are differentiating towards a blood cell lineage, and CD34 is a marker for progenitor stem-like blood cells. Within that population, we also looked at CD45 and CD90 expression, and we found the lines had between 13 and 22% of the cells also expressing those markers within that first population. So CD45 is a marker for nucleated blood cells, and CD90 is just an additional marker to kind of assess the HSC enrichment of the population. We also expressed transgene expression, and we saw minimal losses as well after undergoing differentiation into HSPCs. So specifically with our SMAR modified lines, comparing this with iPSCs that were kept in culture in parallel while another subset was undergoing the differentiation, we saw around a 6% drop, so very minimal. So going from nearly 100 to around 94%. And we also saw a drop in the intensity of the transgene expression, which was expected. We also saw this drop as well with the lentiviral vector modified. And when we looked at the percentage of cells retaining transgene expression, we also had a drop from around 78 to 67%, so about 10%. To then get an indication of the functionality of the HSPCs we just produced, we did an in vitro functionality assay using methyl calt. So for that, we plated the cells in special medium conditions. And if we had a particular blood progenitor cell, we would then be producing a colony from that. So in this case, if we were to have a CFUE or a BFUE, this would be an indication that this colony was produced from a urethroid progenitor cell. With the CFU GM, indication it's produced from a granulocyte and macrophage progenitor and then the CFU GEM is an indication that we have a multipotent progenitor cell because we essentially are having a combination of these different colonies. So we found with the SMAR DNA vector modified lines, we had a larger variety in these colonies, more so actually than even non-modified, and even less with the lentiviral vector modified line. So we've shown that it was possible to take these iPSCs and differentiate them into HSPC-like cells. The next step now is T-cell differentiation. So for that, we were using a different kit to produce our HSPCs, and then subsequently the T cells. And this was the stem diff T cell kit from Stem Cell Technologies. And here, this is our lines that underwent HSPC differentiation using embryoid bodies. And looking at CD34 expression, we found between 25 and 60% of the cells expressing that. And actually with our modified lines, we had the highest percentages of that. I also have additional markers here that you can see here visually, but I would like to point out they're not so important because at this stage, we really only care about having a progenitor cell that will differentiate into a T cell. So the stemness of our populations and whatnot, it's not so important. So in that case, we also looked at CD33 expression. So CD33 has been shown to be expressed on myeloid cells. So because we want to produce a T cell, we would, in this case, not want to see a lot of CD33 expression on our cells because it would be indicating that they're now primed for the myeloid lineage rather than the possibility to differentiate through the lymphoid lineage. So we actually did find minimal expression of CD3 on these cells using the different lines. Assessing transgene expression, we actually saw larger drops when using this EB method to do this differentiation. So with our SMAR DNA vector modified lines, we went from around 100% to around 60. And with our lentiviral vector modified lines, we went from around 91% to 27. And we can also look at the intensity again with our microscope images. And we can see also using this method, we had a higher intensity using the SMAR DNA vector modified cells. So I then took this control line and we continued the differentiation into T cells. So these are the control cells that underwent the T cell differentiation for this protocol. And we found above 60% of the cells were expressing CD45 and CD7. So CD45, again, is just indication that we have nucleated blood cells. CD7 is an early marker for T cell development. And then within that population, we had a high percentage of cells expressing CD4 and or CD8. So at this stage, we would expect double positive. So unfortunately, I do not have data for the modified lines due to technical issues. Yeah, it was my first time going through the protocol, so still getting comfortable with them. But I was able to manage to continue with the non-modified, and I'm really happy that I could share this with you right now. So all in all, we've been able to produce urinary-derived iPS cells. We have an indication that yes, a proof of concept that yes, SMAR DNA vectors can be used to genetically modify these cells and differentiate them into HSPCs and HSPCs that are indicated to produce T cells as well. And we've screened various vector constructs to give us the best success for that. So obviously there's more data that needs to be done. And I'm personally really excited to see what the future brings for this project. So I would like to thank everybody in my lab. Yeah, so everyone listed here who's helped in some way, people that also aren't a part of my lab or our lab and helped out with the project. And I would like to give a really big thanks to all the attendees who took time out of their day to hear me present our lab's research. That's really cool. And thank you so much for giving me this opportunity to share with you.
Yeah, thank you very much, Julia, for this really great and fantastic presentation. I think I was really amazed to see the progress in this SMAR technology and that you are able to maintain the plasmid or the vector in fast dividing cells like iPS cells and T cells as well. We have quite a few questions that came in, but I would like to start with one or two of mine, actually. I was wondering, do you have any idea how many SMAR vectors you have per cell?
Yeah, that's a really good question. So we have done data on this within our lab. Personally, I haven't, but other members have. And we've shown between 1 to 2 vectors that are persisting.
I saw that in some of your flow cytometry plots that expression can vary quite a bit. I think in some of the plots, it was over 3 log scales.
Yeah.
Is there an explanation for that? Are the vectors lost or are they silenced, or what happens that you have a 1,000-fold difference in expression levels?
You mean for the transfection efficiency data?
I think you showed TD tomato expression in iPSCs. I think this is what I'm referring to.
Okay. But the transfection
before sorting, I think after sorting it was much more uniform.
Okay.
Yes.
Yeah. So before sorting, when we first introduced the vectors into them, I don't want to say this like concrete because obviously we need data to support that. But my personal hypothesis is that we have a large variety in the number of vectors that are present in the cells. And then over time, while we're collecting that subpopulation that's actually maintained the one or two vectors, we have a large variety at first before we get to that.
Okay, good. I think I start with some questions from the audience, and one is, could you use a second SMR vector to push cells in one specific lineage? So could you use a second vector for differentiation, actually, to induce differentiation?
Yeah, so we have something kind of similar but reverse. So reprogramming, we've used our vectors to try to reprogram iPSCs, and we have data that shows that we were able to do that. There's a paper that we've published in our lab on that. Particularly for differentiation, I'm sure you can, just depends on what genes you need to be expressed. I mean, it is a genetic modification tool that one can use. How exactly you go about that, I don't really know. I'm not super expert, but if you need a particular gene expressed, you can for sure use SMR vectors.
And this was the question I mentioned before was from Sarah. I think the question from Jamal is about the integrity of genomic integrity. Yeah, have you any ideas about how stable these urine-derived iPS cells are in terms of genome integrity?
Yeah, so this is a really good question. So yeah, it's very important that we're making sure that we do not have integration into the genome because we're claiming, that the vector is persisting episomal. So we, as a group, are working really hard to get this data. From my understanding, it's been a little bit difficult, one, because of costs, and two, we need to look for really rare integration sites, and we don't have an idea in the genome where we could have those, where there's common integration sites for a lot of viral vectors, or if you're using CRISPR-Cas, you kind of have an idea for that, but in our case, it's just could be anywhere. So we've been working on that. It's just, it's also very expensive. Yeah.
Jamal also wants to know differentiation efficiency into HSCs and then T cells. Do you know how many cells from your starting material and when you end up with T cells? What is the efficiency? Do you have an idea?
Yeah, so I've only conducted the protocol a couple times, but we did have issues with getting a large number of cells. So we did have a very small percentage of cells from our starting population after the HSPC differentiation, but I'm not going to say that's because of the protocol. It could also be because we've only done the protocol a couple times. But yeah, I would say there was a small number. And if we wanted to do an upscale for a clinical application, it's something we would really need to focus on to try to improve.
I think maybe a follow-up question. Sai Sang wants to know whether this was a stepwise differentiation process, that you go from iPSCs to HSPC and then to T cells, or once you start the differentiation, it rushes through?
Yeah, so you first go into HSPC and then you differentiate to a T cell. So when we were doing the last part of my presentation, we first had to assess HSPC markers. So we were looking at CD34 expression and seeding the positive CD34 cells, which is a marker for HSPCs. And then we continued differentiation into a T cell using those cells.
Can you keep them in this hematopoietic stem cell state?
Yeah, that's a little bit difficult. So for mouse HSPCs, there is a method that's been published a few years ago where you can keep them in culture for quite a long period of time. But for human, it's I would personally say there's not really a standardized protocol for maintaining them in culture very long. Usually when you're going to do electroporation of human HSPCs, you keep them in culture for a short period of time and you try to get them into the patient as quick as possible.
I think Fabian wants to know, do you see or do you foresee a way to induce clearance of SMR plasmids from cells at a defined time? So if you want to get rid of them.
I like your idea, Fabian. I personally, yeah, that would be a really good PhD project. I don't know how this at a genomic level this would work, but I'm sure someone could find a way. I just understand that when we're using the SMR vectors, once they're established, they're maintained. I mean, if I had to think right now, if there were ways to silence it, that definitely, I think there's techniques to do that. But to remove the vector from the cell, I don't know exactly how that would exactly go.
Your PI come up with a novel PhD project.
Yeah.
Some people want to know, I think you showed that you can differentiate urine-derived iPSCs into different lineages. Some people wanted to know exactly the cell type as well. One question from, let me find it. Marcus wants to know whether you can differentiate them to cardiovascular cells.
Okay.
And Izatu wants to know, where was it? In epithelial cells. Have you done this?
So epithelial cells, from my understanding, we haven't done this in the lab. In principle, when you have an iPS cell, you should be able to essentially differentiate it into any human cell type if there's an established protocol for that. Particularly in our lab, we've used it to differentiate them into kidney organoids. And for cardiovascular cells, yeah, we haven't had the aim to differentiate them into the cell type, but we actually found within some of our differentiation protocols to produce HSPCs, we had cells that were beating like a heart in our dishes. So that was kind of cool, but we don't really have any phenotype for that. And just visually, we saw this present.
Follow-up question from Izatu about the impact of the genetic background on differentiation propensities, or maybe also how good they are to actually be converted into iPSCs. Do you have you done this for different donors?
You mean like?
I think that's the question.
Yeah. Yeah. So this is something also really like karyotyping and all that stuff. Mm-hmm. That's really important to do. We need to make sure that the cells are maintaining, they're not being altered in a way that they're prone to become a cancer cell. We don't really, this project that what I'm showing here is more of a proof of concept. We have a lot of data lacking more on the safety aspect of that, but it is really important. And when you have iPSC lines, it's just classic method that you're analyzing them after certain periods to make sure that they're maintaining a normal karyotype and things like that.
But have you produced iPSCs from different donors? I mean, collecting urine from different people, and then did you see differences in the iPSC production efficiency, and then later on also in differentiation?
Yeah, so for this project, we were using the cells primarily from, we were using the cells from one donor, but we did have two in our lab that we've used, and we did see a difference in efficiency when using this other donor compared to the one we're using now. So yeah, it's normal to see differences in the success. Also, you can see differences within colonies. So from one donor that you're using to engineer iPSCs, you will have multiple colonies from that, and you can find differences between those colonies that were produced in the beginning.
Great. I think in terms of application in the future, I guess GMP grade, iPSCs, and so on and so forth. Unknown or anonymous wants to know, can the transduction and the differentiation be done in an automated closed system, such as Prodigy?
Yeah, that's yeah. So we've done this. We've done this with T cells, primary T cells. We've used the Prodigy for genetic modification using our SMR vectors. So yes, with that, I don't have doubts that it could also be possible with an iPS cell as well. It's just the way we're modifying them right now in the lab is using liquefaction, but I don't see it being. It's been shown before that we can do this with other cell types, and I don't see why not with an iPSC.
I guess the last question now from Iman. How hard is it to culture the vector-modified iPSCs in culture for a month? Or maybe also how stable is the SMR vector in iPSCs? So
once you have it established, it just, what we show is that it's maintained. So this is data actually that we're missing. We've tried to do this, but yeah, it takes so long to establish and compare all the features. But I really would have liked to have had data where we're looking at the retention of our vectors in the iPSCs long term. And that's still an experiment we need to do and really assess. But what we've shown right now is that when you have the line established, regardless of comparing features and whatnot, once you have that line established, it will maintain long term, for at least the period we've been using the lines.
Okay, great. I think we are through all the questions I can see on my screen, and we are perfectly in time. So thanks a lot, Julia, for this really fantastic presentations and also for getting through all these questions. I think this was a great event. I would like to thank you one more time. And of course, also the people online, also for their questions and for this lively discussion we had. Thanks a lot in the name of the German Society of Gene Therapy, and enjoy your afternoon. Bye-bye.
Bye, Tony. Bye, everyone.